I'd like to welcome all of you to the STEM Speaker Series. A little bit about it. The STEM Speaker Series is held on the fourth Tuesday at 11:10 a.m. during the months of October and November, and in the spring semester, February, March, and April. So there are 5 talks altogether. This is the second and last talk of the fall semester. The next one will be next February, 4th Tuesday, heavily advertised with posters all around campus and by email to all faculty. To whet your appetite for what's coming up, on Tuesday, February 28th, our first talk of the spring semester, a representative of the Media Police Department Forensics Lab will give a talk on how to use science to catch criminals in Delaware County, which should be rather interesting. In March 26th at 11:10, Alan Green of Michel Clusiel Chocolatier, Paris, will give a talk on the art, science, and medicinal benefits of chocolate. And April 25th, next semester, I am going to give a talk on President James Abram Garfield, one of our presidents who was assassinated, and his discovery of a new proof of the Pythagorean theorem. So President James A. Garfield as mathematician. So those are the talks coming up in the spring. I'm going to circulate a roster, which I will scan later this afternoon and email to all faculty as evidence that you attended this talk. So I'll start it here. It should go along each row, then to the row behind it, all the way back, and this side of the auditorium. And eventually it needs to make it to me by noon, because I've got to run and teach a class. So please put your name, and next to your name, the professor for whom you're going to get credit, or extra credit presumably, and the class. So I'm going to start it here with Catherine, and it should circulate on this side of the auditorium when it makes it back there to the other side, and eventually up to me, because I do have to leave at noon to teach a class. So our talk today is the race for the discovery of DNA, our own Dr. Bela Dadhich, who has taught at Widener University, University of Pennsylvania, Cabrini College, and Drexel University, and we're so fortunate to have her here teaching biology at DCCC. I could spend the entire 50 minutes talking about all the teaching awards she's won and all of her publications, but I'd rather give time to her. So please welcome Dr. Bela Dadhich.
So I'm going to start today. Oh, thank you so much for that. Very nice. I'm going to start today with a topic that has fascinated me since I have worked on my PhD. So I didn't really have the background of truly the study of the structure, but I did work on the replication of DNA when I was doing my PhD at Drexel University. We worked with the DNA of bacteria in E. coli. And so when I was working in the lab, there would be like a lot of free time where you would read other publications and you'd spend your time doing some research on other findings. I came across a couple of papers that dealt with the discovery of the DNA structure that truly fascinated me. And from that day on, I literally feel like I'm obsessed with this story. Every time I study how this structure, every time I look at anything related to DNA structure, it's as if I've learned the story for the first time. I get deeply engrossed in it, and I learn a little bit more every single time. So finally, after talking to Dr. Holbus about this story multiple times, I think he said, why don't you just do a seminar on it? I said, that sounds great. And so today I'm going to present to you a little bit of the historical perspective that came about about one of the most important discoveries, truly, in science. And so, really, it's, if you look at science, the way science works, is if you learn the structure of important molecules, it segues or leads into understanding the function of it. And so this era is around the late 40s, early 50s, when they knew of this molecule called DNA, that it was very important in terms of inheritance. They just didn't know how it did that. They knew it was important. They knew it had some sort of hereditary component to it, but they just didn't know how. And scientists always want to know things. That's their big thing. They want to learn. They want to learn about life. They want to learn how things operate, and they don't settle for not knowing. And they think that's okay. So they knew at the time that this was important. We really need to figure out the structure of DNA. And there were a bunch of scientists working on it, and they knew whoever would figure that out could truly figure out the secret of life. And they would become golden. And the story takes place in that era of scientists racing to the finish line of understanding this really important molecule. And so I just want to tell, I think I see a lot of my students here, they know what DNA is. So just to give a refresher of it, so you see what we're looking at when we're looking at this perspective of figuring it out. DNA appears as a double helix, helical form. It's got sugars in it, it's got phosphates in it that create literally the backbone you see here. And then in the middle of it, there are these nitrogen bases, and there's 4 of them, and they're represented as the letters A, T, C, and G, which truly is considered the alphabet of DNA. So that's how they have it. And basically, the base A pairs with the base T, and the base C pairs with the base G. C and G go together throughout the entire length of the DNA. That's how it's organized. And we all study this, and we teach it in biology, and everyone learns it if you're taking a biology course. But what we don't realize is all the scrutiny and effort it took to figure out this structure. It seems so easy now, but be in that era and you will really realize how difficult it was and what they went through in order to figure this out. All right, so there were 5 key players in this race to determine the DNA structure, like what I just went over. They didn't know it was a helix. They didn't know that A paired with T. That's what they were trying to figure out. And these were the 5 key players. Francis Crick, James Watson, a lady by the name of Rosalind Franklin, Maurice Wilkins, and Linus Pauling. Those were your 5 players. And so we see the interplay in this discovery that involved these 5 major scientists and how it all came about in this lecture. So these were the 5, just to give you a little bit of idea of where they were. Linus Pauling was in California, United States researcher. Maurice Wilkins, Rosalind Franklin both worked at King's College in England. Francis Crick was in England as well at Cambridge University. And James Watson was in Chicago at the time. And you'll see he travels and enters into Cambridge University because you'll realize that he wants a part of this. Because this discovery was so big time. And if you look at their biographies that have been written, they had only one priority in mind, and that was to get to the finish line and be the first to determine the structure of DNA. Because they knew if we did that, we would be golden. That's it. The world is ours. And from that one discovery, they will be able to make such a great impact on human life and understanding how it exists, how inheritance happens, how new forms of life form. It would just give you the best idea of all these processes. So it was big time. I mean, this was high level stress at this time, all 5 of them. Basically, they thought of nothing else but DNA at the time. So let's talk a little bit about Linus Pauling. Linus Pauling was literally brilliant. He was a genius. He was considered one of the smartest people truly in the United States at the time. He won 2 Nobel Prizes. He was a chemist, and basically he was an expert in model design. Give him a bunch of molecules and atoms and he can create the best model of it. And somehow he had tremendous insight that it always was right. Whatever he figured out actually was what the molecule was. He'd be like, this is what it is. They did the research, that's exactly what it is. He was brilliant. And he stated, you know, DNA is important. I think DNA is a triple helix. And people listened to him because he was Linus Pauling, the master of model design. He was amazing. All right, so that's him. The next 2 important researchers in this game of DNA structure were 2 researchers at King's College. They worked in John Randall's lab in England. And basically, it was the lady I mentioned, Rosalind Franklin, and Maurice Wilkins. And both of them used this special technology called X-ray crystallography, which we'll talk about shortly, which is a technique that requires skill and intense determination to use. But if you can use it and you've got that skill and that determination, you can figure out the structure of any compound out there. And both of them were highly trained in the use of that technology. And in fact, Rosalind Franklin, as we're going to get into, she created some beautiful pictures of DNA. So she was very well skilled in using this technique. A little bit about Rosalind Franklin. This is another child when she was born. They were like, wow, this girl is special. I mean, she just gets everything. She's so intelligent. Actually, Sid, you'd love her. Her fun pastime game was arithmetic. She just loved math. She constantly tried to figure out math games. Her friends would come over and they'd be like, what do you want to play today? What do you want to play? Shall we get our dolls out? And she goes, no, let's play math facts. And see who's going to be the first to get all the facts done. And of course, she was the one who won. Her parents knew this girl is so bright, so intelligent, we have to really put her in the right career path. And she told her parents what she wants to do is do science, because she said science will make a difference. She goes, if I have a discovery in science, I will make a difference to mankind. And she really felt that. Many times her dad was like, no, I don't really think I want you going into this man's field. What are you doing there? You know, they're not gonna let you succeed. So her dad was, actually her parents were very nervous about it, but she fought her way. She told her dad, that's what I want to do. I want to go in research. I want to do science. And she had great backing from her mother, who really said, we're going to promote her. We're going to let her achieve what she can achieve. And she literally was known in the international community for great skill. Her hands literally were thought of as gold. Whatever she touched, she perfected. And she had become this international sensation. Like, she got well known for that high skill that she had. She really could achieve anything. Once she said that she wanted to do it, she did it. And she did it with perfection. And so, again, very well known. So John Randall invited her to come work in his lab. He was a pioneer. He was a leader in this field of research. Everybody knew. Randall's lab is working on DNA structure. So everybody knew they're going to be at the forefront of this research. And he hired her because he knew she was the expert on X-ray crystallography. And so she went to his lab and she started working on it, X-ray crystallography of DNA. And so I want to show you a little bit of what, keep saying X-ray crystallography, X-ray crystallography. What is it? It is a very sensitive technology. So it has multiple parts to it. First of all, you have to have the ability to crystallize the molecule or the compound that you want to determine the structure of. That in itself is a very tough task because crystals fall apart. And so you have to have great ability to manage these molecules so perfectly that they stay stable. And she had the ability to do that. So she created crystallized, perfect molecules of DNA like no one else. I can tell you, even with our technology today, we can't do what she did. That's how perfect her technique was. And so she created these crystallized molecules of DNA. Next step is you shine an X-ray beam on the molecule. And that requires skill as well. Because if your beam is too thick, you get a very dark background. And you're not going to decipher any results. If it's too thin, then you're going to see a very faint background. And then that's not going to work. So the beams have to be perfect. And she had, again, the perfection in technique. She knew exactly what level the X-ray beam had to be. That would basically shine on the crystallized perfect molecule, which would then scatter light onto a film based on what type of structure it was. Every molecule, based on their structure, gives you a different pattern on your film. So that's what she did. And then when you get your pattern, you use a bunch of mathematical equations and analysis to then figure out what that scattering meant and what type of structure you were dealing with. And so that's what she did. She created the X-ray photographs of the DNA molecule. So that's what her job was. And then to get the picture and then to analyze it. The picture is her data. She analyzes it, and then from there, you determine the structure of DNA like we all have learned is a double helix. Now, she was working with Maurice Wilkins in the same lab. He started before her. Before she was hired, he was working in the lab, and he was working as well on the same technology, the X-ray crystallography, but he just couldn't get the best results. He couldn't do it like her. His results were fuzzy, there were so many discrepancies, his crystal molecule fell apart. He just could not get the results that she got, and he majorly resented her for that. He was always angry. He had a very negative approach towards her. He never wanted to collaborate. He didn't want to have any discussion with her. He literally was hopeful she wouldn't even be in his lab. That's how he treated her. She was always, basically, this is her best friend. Her best friend wrote a biography on her. And she said, always very mean to her. I mean, it was just a very negative approach he had towards her. And he really, when she got hired, in the lab, he really thought she was his technical assistant, a mere technician that was supposed to fetch different items for her as she needed a solution, she would go make it and deliver it. He never really treated her as his equal. And she was like, no, I wasn't hired to be your assistant. We both are on the same level of field here in terms of our position. It wasn't very nice for her. This was the result he got. Very fuzzy results. He always blamed the technology. He's like, oh, look at this machine. If we had a better machine, I would have better pictures. He never took responsibility for his own work. So I was like, yeah, this is it. So at one point, Randall, what happens in research is you want to get your research out there. You go to like conferences and you present your work. Of course, you don't reveal everything, but you reveal just enough so that people know you're working on this topic of interest. And then you establish yourself in the scientific community. Oh, this lab is working on DNA structure. That's big time right there. These guys are big time researchers. So he was sent out to a conference and he presented this work, because you're not going to get anything out of this. Some of his fuzzy pictures he presented, but at least he got the word out there that John Randall's lab is working on DNA structure, and that's big. So, as he's presenting this, a man by the name of James Watson shows up to that conference and is fascinated by the work. Now, James Watson, my friends, is himself a total genius. He got his bachelor's degree at the age of 50, and he got his PhD at the age of 22. Exactly, and no matter what you gave him, he figured it out, and his bosses couldn't keep up with him because he was just so smart. And so they're like, what to do with this guy? Send him to this conference. Get him out of my hair. And he goes to the conference and he sees Maurice Wilkins' data on DNA structure. And he's like, man, this guy's made no progress. I need a piece of this. So he's like, I got to get into DNA research. Because he knew that that's whoever figures out the structure of DNA is going to be big time. So he's like, I want to be famous. This was his autobiography. Quoted from there. He always wanted to be famous. He wanted to make such a major discovery that students like you guys taking biology would know his names. That's how he wanted to do that. He wanted to make some major discovery. So he said, I want to do DNA work. I think that's what I'm going to get out of my micro lab. I'm bored with it all. And I'm going into DNA work. I want to figure out the structure of DNA. Well, he knew he's not getting in John Randall. They're so ahead of the game, they're not letting anyone in now. There. So he starts looking at which schools in England, which research labs are working on X-ray crystallography. And he realizes that there's a Nobel laureate's lab whose name is Braggs. And he contacts him, and he also knows that that lab is working on X-ray crystallography, but more so proteins, not really DNA. So he says, I need to get in that lab, and I need to understand and learn that technique. And then I can hopefully learn it quickly, because he was smart. He's like, I'll learn it very quickly, and then I'll get into learning how to do the structure, figure that out. So he gets to the lab, Braggs accepts him. I mean, he's very smart, but he gets in. And then Braggs says, okay, I'm gonna have you work with this guy, Francis Crick, because he is our X-ray crystallography leader. He's our leader of that. And the two of them get along great. Watson and Crick instantly hit it off. They're on the same page. They become like best friends. So this is Francis Crick. Francis Crick as smart as he was, he couldn't get things done. He was always delayed in everything. He continually changes his goals. He looks for the next big thing. He just is unable to get the project at hand complete. And another point about him, he's very good friends with Maurice. That was his word. Very good friends. And so Watson and Crick come together, and they sit down. They're like, all right, let's have a game plan. They're like, all right, we need to figure out the structure of DNA. So we gotta learn X-ray crystallography, then we gotta learn how to crystallize DNA, and then we gotta learn the technique, and then we gotta get this picture, and then we gotta do all this work, and then we gotta win. And they're like, well, those guys are way ahead of us. They are like almost there. And so they decide, you know, maybe let's not do that. That's too much time. Instead, let's just sit around and we'll like look up the research of everybody. And no one's doing this. We'll just collect everyone's research and we'll put it all together and then we'll figure out the structure of DNA without doing one experiment. They said, that's what we're gonna do. And Crick's like, sounds good to me. He didn't really want to do the work. So he's like, let's do it. So they decide, instead of doing experiments, they're going to just collect research articles. They're going to read research articles, and then they're going to build a model of DNA based on all the papers they read. And people literally, I saw this, I've been reading up on this for a long time now. And I saw that this one article that basically was a pretty well-respected article said, they looked at them, the two of them, as a pair of lazy jokers. Because all they did was they went to bars, they drank, and they just talked about papers. And they didn't really do any work. So they were just like, look at these guys, look at these two. Yeah, they're gonna win. They're really gonna get that structure of DNA. So they came up with this plan, and they said, we're gonna build a model. So, Rosalind Franklin, one of the other things that happens when you do research at a university setting, or any place where they have research, is you do your research and then you have these seminars that you present. Like every lab presents a seminar. Sometimes it's once a month, sometimes it's every week. And there's this seminar series, and it's open to everyone. Anyone who wants to come and present. So Rosalind Franklin, it was her turn, and she was presenting her work on DNA, and Watson happened to go to it. Because Wilkins told Crick she's going to present today. And Crick does X-ray crystallography, and so Crick said, let's go see this lady. She's been working on DNA. Maybe we can get something out of it that'll help us in our model. So let's go in there and hear her speak. And she presented, and they all treated her so badly. They sat around like they were like heckling her and reading a newspaper while she was presenting because they wanted her to feel like no one cares what you're doing. You think you're big time, you're not. And don't forget, you're a lady. So they really weren't very gracious. They were actually extremely rude to her. But Watson was smart. He still knew she does know something about DNA. So he's like, what I'm gonna do is I'm gonna build my model, and I'm gonna invite her, and I'm gonna have her take a look at the model, and then maybe she can guide, like just mention to her, hey, we just wanted you to see this. We're not really looking at your opinion, but, and then maybe from there we can kind of get an idea of whether we're on the right track. And so he decides to, Watson and Crick really, Watson decides to show her the model. And she basically laughs at it. She goes, that's your model of DNA? Because she's been studying it. She's been seeing pictures. So she kind of has an idea, but she just needs that best picture. And so she's like, that's your model? She goes, and laughs at it. So Watson gets so upset. Oh my God, she just laughed at me. And she's a lady, and I'm Watson. And what's going on? And he basically created a model with the bases on the outside. He's like, all right, there's these 4 bases, A, T, C, and G, and they're on the outside. And she's like, yeah, that's not your model. And it ends up becoming an embarrassing failure, and Braggs actually gets really mad at the two of them and says, I need you in the lab starting to do some research. Because I don't like being made fun of. I'm a Nobel laureate, and no one makes fun of me. And you two are bringing me down. You guys gotta do some work. So end the modeling and start getting in the lab. So he was very upset. So meanwhile, Franklin, however, is really happy. Because this was a quote from her, let the data talk. She was a traditional biologist. She said, you don't make up stuff. You do experiments, you collect data, and your data is your indicator of what is going on. You gotta sit down, you gotta do the hard work, you gotta do the labor, you gotta have the ideas, you gotta put them in place, you have to collect the data. And when she saw the failed model, this further confirmed her, so she knew she was on the right track. She knew she was doing it right. And she wasn't just like, oh, I got any picture, let me use it. She was like, no, I'm going for the best picture, because that's where I'm gonna get the best data. And I have to do a good job, and it has to be. And that was her idea. She never wanted to do anything just to do it. She had to do it right. And so she said, this is hers. Using other people's data and model building is just not good enough. You actually have to get your own. Or you have to be a good collaborator so that people will work with you and show you and share their data. You can't make fun of people and bring them down if you need their data. You have to be a good collaborator. And so in 1952, she takes this picture of DNA, one of the most perfect pictures of DNA you will ever see. Even with the technology we have today, there is no better picture than this picture she took of DNA. And when she got that result, she knew right away she's got it. She has something special. And she's ready to solve this problem of DNA structure. And she's there because guess what? Nobody else has this picture. Only she does. And so when she saw that, she goes, I think I'm going to win this thing. I think I'm really going to win this thing. Who's got this picture? And so she named the picture Photograph 51. Like I said, it's the most perfect diffraction image of DNA to date. It's the sharpest image, and it has the signature X, which is classic double helix. And you look at that, and you can, with mathematical analysis, you are able to figure out where the bases turn and how the helix is organized, and she has it. And she's so happy, but she's still like, I have to analyze the data. I'm going to have to find the mathematical algorithms to do this. A lot of people aren't really talking to me. They're not collaborating with me, but guess what? I've got the picture, and nobody else has it. So she's got this picture, and basically Wilkins, his mode of operation was he's just going to continue to present. He's just like, I can't take this lady anymore. I can't have her here. I can't work with her. It's just not working out. And Watson actually tries to intimidate her. So he ends up going to her, and he's like, because he knows she's got pictures. And he's like, I need those pictures. And so what Watson does is he's like, I'm going to take the approach of being very rude. And maybe that will work. I don't know why he thought that, but he said, if I'm like really mean to her, and even more rude than I have been, maybe then she'll give me the picture. And so he goes to her and he tells her, look, you need to give me the picture because you don't know how to analyze data. I know how to do it. Just show me your data. And she's like, get out of my lab. You need to leave. And so he leaves. And so he's like, now what am I going to do? And Wilkins says, Watson, you want to see something that I think you'll really like to see? And Watson's like, what? And Wilkins, without Rosalind Franklin's permission, shows Watson the picture. published DNA results. He says, look, I got a picture to show you. Maybe this will help you with your model building. And she didn't know. He pulled the picture out like real quick. He said, I can only show you this really quick. And he pulls the picture out and he shows it to her. And this is a quote from his autobiography. The instant I saw the picture, my mouth fell open, my pulse began to race because he knew he had it in his hand. The answer to the greatest mystery of the time. The secret of life was about to be solved. Races over to Crick, and he actually jots it down. The guy was a genius. So he basically could have the photographic memory. He jots it, they didn't even have a paper on him. He jots it on his newspaper. And he runs over to Crick, and he's like, Crick, I think we got the data we need. And they sit down together. Crick was an expert in X-ray crystallography. He knew what the signature X meant. And the two of them sat down and they built the entire double helix molecule using her data without her knowledge. They figured it out. It's a double helix. And they realized from the data, the bases aren't on the outside, the bases are on the inside. And Watson, the genius he was able to remember all the research that he was reading, and he remembered there was this one paper where this guy by the name of Chargaff did some research on DNA. And Chargaff studied DNA, and what he noticed was no matter which DNA he looked at, from any organism, bacteria, fish, human, plants, whatever DNA he analyzed, the A's always equaled the T's and the C's always equaled the G's, but he didn't know what it meant. And he published the paper. He's like, I don't know what this means, but every time I look at DNA, A's equal T's and C's equal G's. And Watson was like, remember those Chargaff's rules? And guess what? Inside, that's how the bases are here. A with T and C with G, looking like that. And based on the picture, they were able to determine the turns of the helix and create the model of DNA. Figured out the puzzle from photograph 51 that they acquired without the knowledge of the lady who actually did the work to get it. And so they become the winners in the identification of the DNA structure, and they make their model. They actually show Rosalind Franklin. They're like, you know how you laughed at our last model? How do you like this model? And she didn't know that her data was used, but she looked at the model and was like, yeah, they're right. That's it. That's your DNA structure. And she told them, you're right. That's right. And then she actually was writing a paper, and she ended up they have it, handwritten by her. She crosses off the section that she had written, and she said, as deduced by Crick and Watson in handwriting. She didn't know they took her work to figure it out. And then these Watson and Crick, they decide to publish the paper in the journal Nature, which is one of the highly respected journals in science. It's so hard to get a publication in there. They knew this was big time. They're like, yep, we're getting a Nature publication. They write to Nature. We're going to publish this. We don't need a whole lot of room here. Give us one page. And they write it up, and they don't give her credit. They don't even mention her name. And in fact, they say if someone else out there has any evidence for this, we're unaware, even though they took her picture. And so that's how they said it. And then John Randall, however, was very upset because he's like, we've been working on this for years and years. And these guys, what did they work on it? Less than a year? And they're publishing a paper on it, and they have no data, and we've been doing all this work. So he actually was famous. So he called up Nature and said, this isn't right. We've been working on it. You're publishing their paper. We're going to get no credit here. We've spent millions on this. What have they spent? And so Nature says, okay, we're going to do Watson and Crick's article first. We will do Maurice Wilkins' paper second, and Rosalind Franklin's will be in the end, and her paper simply made it seem like she just confirmed their results. And that is how nature did it. Watson and Crick got all the credit, and they also won the Nobel Prize for the discovery of DNA in 1962, along with Maurice Wilkins. The three of them won the Nobel Prize for the discovery of DNA. And have you noticed Rosalind Franklin's name is missing from that list? She didn't win the Nobel Prize because guess what happened to her? She ended up dying at an early age from ovarian cancer that they attribute to her high exposure to X-ray beams in working on the study of DNA, because she was so engrossed in the work that many times she didn't have time to protect herself from the radiation. She didn't want her DNA to fall apart by running over to get embedded. She was so involved in the research. She never got any recognition for the great impact she made on this discovery of one of the most important molecules in the history of science. And from this discovery, they've discovered so much more. Every discovery came out of that. A lot of what we know about genetics came from this major discovery. And you know what's funny about her, Rosalind Franklin? After she left this lab, like she had decided after that hostile environment, and she's like, I'm gonna analyze this photograph and then I'm gonna leave. And she was very good. I mean, she had multiple job offers. She ended up going to a lab that was working on the tobacco mosaic virus, and she ended up figuring out that virus, as well as basically doing research giving us a great understanding of the infectious nature of viruses, that she could have won another Nobel Prize for that discovery as well. Every place she went, she made a great impact. And her discoveries were big time. As I say, her papers are still quoted today. So that is why I'm so fascinated by the story and tell it like it's the first time. You should see my family at home. They're like, look, don't tell us that story again. We've heard it. Because I'm so fascinated by it, and I hope that you have enjoyed it as well. Thank you so much for listening.